Thanks, Vivi. Let's get started with the project. Scheiße, with the team introduction.
I would like to introduce
FAU-GEM.
Hello and welcome to FAU-GEM.
Hello, I'm Maria and I'm today's host, and sitting next to me is Vivienne.
Yes, hello.
Vivienne is going to introduce you to today's program.
Yeah, so welcome guys to today's episode. Today we will start talking about a new team, which is called the UteRus team. And I will shortly introduce the team members to you, where they are from, what they are doing. Then we address the problem they cope with, and guide you a bit through their project. And in the end, we will also discuss a bit the collaborations they were taking part in, and talk about the related teams or team projects which exist in the iGEM universe.
Thanks, Vivi. Let's get started with the team introduction. The team consists out of 12 members. It's an undergraduate team, and what's special about this team is that they got started with their project in 2020, right before Corona. And then when the pandemic hit, they weren't able to access the laboratories.
Wow, that is crazy. I mean, they had to kind of get around all the lab stuff. How did they do that?
Yeah, exactly. They couldn't access the lab, and that's why they had to shift their project focus. And what they did was that they started to develop certain wet lab protocols and also focused on designing hardware, software, and
so they took more of the computational part then.
Yes, they have a great dry lab part in the wiki. You should check it out because they did a lot. And also they focused on human practice and education, and started to even learn sign language to communicate their project to the public, and collaborated with with foundations that were specific to the topic we're going to discuss today. Yes, and it's no surprise that they won many awards because of that.
Of course. It sounds like a lot they have done.
Yes, and that's why they won a gold medal. Also, it's a diagnostic team, so they won Best Diagnostic Project. They won an Inclusivity Award because of the sign language stuff they did. And they were nominated for Best Software, for Best Integrated Human Practice, and also for Best Education.
Very, very interesting. Thank you for introducing the team to us, Maria. I think now that we talked about what a lot of prizes they won, we should also maybe address which problem they dealt with. Like, what is the project?
Yes, I almost said it already, but they deal with endometriosis. And what they wanted to do was to create a novel diagnostic device for endometriosis, and they did that using the menstrual effluent. Surprisingly, their team name also, it's called UteRus, refers to the female reproductive organ. And for those of you that don't know what endometriosis is, Vivi is going to introduce you to that problematic.
Yes, I will explain the syndrome a bit to you. Of course, the male population listening here is maybe not very familiar with that, so I think we will definitely lighten some paths here. So endometriosis is actually a chronic disease which is affecting around 200 million women worldwide, and the symptoms progress also over the time. So actually, 1 out of 10 women is affected by endometriosis. And what does it do? Yeah, it's it causes abnormal growth of endometrial-like tissues outside of the uterine cavity. So a lot of women get these tissues in, for example, the urine tract region, but also in the gut or outside of the ileum, and it really causes a lot of pain also during the periods, because those tissues tend to also kind of menstruate in that time, but as they are not part of the uterus anymore and are outside, there's also pain in different regions of the body, which you can imagine is not very funny.
Yes, it sounds very unpleasant.
Yeah, and it can also lead to infertility, which is even more shitty. Sorry for my language here. But it is really hard. And the worst thing about this is that it is super hard to diagnose. And more than 11% of the patients are undiagnosed and suffer like 7 years before one doctor is finding out of the problem. So a lot of people went to more than 5 doctors until they are diagnosed with it. And I think this is actually not how it should be. And also sometimes women who want to get pregnant and don't know that they have the problem really try hard to get pregnant for a long time until they found out they cannot get children.
Yes, that's very painful for the patients, I think. And also another problem is the diagnosis, right? Because it's really hard to definitely diagnose it. And I think the only way to really do it is through invasive and expensive exploratory surgery. I mean, there are pelvic exams and ultrasounds and MRI that can be done to confirm the diagnosis, but I think the way to do it is right now the surgery.
And it's very invasive, yeah.
Yes.
And I think, I don't know the English word, but in German it's called Bauchspiegelung.
Yeah, I think it's laparoscopy. Laparoscopy.
Sorry, guys. That's the word in English. Thank you, Maria. And yeah, as you said, it's painful, and of course, it shouldn't be the first way to find out about it. So there must be a creation of a tool where you can get easier access to information like proteins or like a blood test or whatever, to just don't harm the tissue in the first place.
I agree, and that's where our team comes into play. Because before I start introducing the project, maybe we should talk about the motivation for the project.
Oh yeah, that's a good point. Maybe you can do that.
Yeah, I will, because it's very interesting. So they had a team member that is affected by endometriosis. So then they really were able to experience how hard it was for her until she got diagnosed, and also about her symptoms, and it helped them realize about the lack of research that was done, and also the lack of funding, even though so many people are affected. And another thing that inspired them then to continue with the project was that they read a publication about the presence of certain biomarkers for endometriosis in menstrual effluent. And we will link this publication in the description next to our sources, so you should definitely check it out. And also there was another iGEM team from MIT who tried to detect endometriosis using newly characterized parts from mammalian synthetic biology, and this team also inspired them.
So out of this personal motivation, but also the information they found in the current research literature, which project did they form out of that? What arises out of this motivation?
They actually did a lot. So they had a wet lab part, also a hardware part and a software part. I'm going to start with the wet lab part. For the wet lab part, they created a lateral flow immunoassay that uses biomarkers for the detection of endometriosis from menstrual blood. And the lateral flow assay detects the presence of target molecules in the sample using a sandwich-style immunoassay approach. This approach should be known to you maybe by corona tests, because they work the same way. So for this, the following steps are necessary. So at first we have the menstrual effluent that is added to the sample pad. So we have like a test stripe, I think, and the menstrual effluent is added to the sample pad. And this pad contains a fixed concentration of labeled antibodies on a membrane. and antibodies are labeled by conjugating them to gold nanoparticles. The same thing is actually done for corona tests. And then the sample flows, and the labeled antibodies bind to their target molecules, and then they reach a second immobilized antibody at a test line, and this then binds to the target molecule. Other free labeled antibodies without target molecule then continue to travel down the strip and bind to an immobilized antibody
at a control line. So actually, as you said, it's also part of the corona test. That's true. But the fun thing is corona tests are based on pregnancy tests.
Yes.
So actually, this is maybe very funny here because the pregnancy test strategy is the same strategy now for them used for the endometriosis, where you also have like a hormone, I guess, which is catched as an antigen, and here it is the markers.
I think it's really fascinating that the same principle works for different approaches.
That's biology.
Yay!
So yeah, very interesting. And do you know which biomarkers they focused on, or how did they select which biomarkers are appropriate for this?
Yeah, they had a publication, and in this publication there were 12 different biomarkers, and then they selected from these 12 biomarkers 3 with the highest correlation to endometriosis. Mm-hmm. And they did that with the help of log odds ratio. And the biomarkers they then chose were IGF-BP1, which is correlated to altered uterine environment, CA1258, which is related to aberrant tissue growth, and EL6, which is
an inflammation and irritation marker.
Yes, exactly. Mm-hmm. And then they used molecular modeling to identify antibody pairs with non-overlapping epitopes to capture the biomarkers for this assay.
Wow, that's very cool.
Yes, so that alone is a lot, but they achieved a lot. And the project didn't just stop there. They also produced the biomarkers. Oh. And yes, it's they did so much. They created a plug-and-play approach for plasmid design, and then they transformed these plasmids into an E. coli strain. It's called Shuffle, and this strain is used specifically for antibody production. Mm-hmm. And then they created biobricks for variable and constant regions of therapeutic antibodies, and so other people can use it as well. And then with that, they created this novel plug-and-play method. That's how they call it. And as I already said, it enables other people to produce monoclonal antibodies as well. And a question that might arise is, how did they obtain the menstrual effluent?
Yes, that's actually a very interesting question. And here the hardware team of our iGEM team comes into play, because they created a menstrual cup which is specifically designed for endometriosis patients, which is just crazy because, as most of you maybe do not know, it is really painful for endometriosis patients to wear the commercial menstrual cups, as the thickening of the vaginal wall and the pelvic muscle fatigue are very common in those patients. And because of that, it's really painful to wear those cups. So
maybe another design is needed for those patients, right?
Yes. And what they took into account here is that they would need a thin wall and inward-facing rim to reduce the pressure of the cup to the wall, and a rounded stem for an easier grip and comfort, and also a bent cup body for easy insertion is needed here, as well as more volume, because they wanted to collect the menstrual effluent for this tool. They just make the cups bigger, so they can take up to 40 milliliters of effluent. Commercial cups usually can just hold 25 milliliters. Yes. So for heavy menstruation, it is way more easier to have more space for the blood.
But what about the usability of these cups? So how do they sterilize them, right?
Yes. They actually designed a portable UVC sterilizer for these menstrual cups to prevent the bacterial growth. Because if you have bacteria in those cups, as you said, the patients can get the toxic shock syndrome, like a very heavy infection, and we don't want that. So
yeah, and I think that's also really suitable for regions where there isn't much water available constantly. So it maybe even makes the cup approachable for developing countries where this is an issue.
Yeah, and also it could be an inspiration for the industry to design this also for the commercial cups.
Yes, true.
So now that we talked about the menstrual cups and them collecting the menstrual effluent, the question maybe arises for our listeners, why we use menstrual effluent for this creation of the diagnostic tool?
Yes, that's a good question. I've asked myself that also, and the reason why they use menstrual effluent is because it's first a non-invasion method of collecting the sample that's needed. Also, since the specimen collection only occurs during menstruation, we don't have a risk that's not that high of hormonal fluctuations. And of course, the effluent then itself has to be treated, so it can't be used straight from the cup. Instead, they have to centrifuge it to separate out the serum of the menstrual effluent. And for this, and that's where the hardware team comes into account, they created an inexpensive laboratory equipment for clinics without laboratory access. And this, of course, also reduces the costs of the project. And they created a DIY centrifuge. And this centrifuge consists out of recycled toys, computer parts, and simple circuitry. And I think...
How funny.
Yes, I think they got the idea because they didn't really have access to the labs. And they had
to use things they have around.
Yes, exactly. And also, then they, in relation to that, had a modeling team and a software team that, like, beside the centrifuge, also created a predictive model that predicts the endometriosis risk in patients using only symptoms and clinical history with about 85% of accuracy. So I think that's pretty good. And then they implemented that model into a software, and I think it's called endometriosis risk calculator. So it should be on their wiki. Definitely check it out because it's great that they did that only like from home. And also another DIY thing they built was an LFA imaging station, where they had just a really inexpensive visual imaging station that they created by using Lego blocks. So again, something that was just probably lying around at home. And then they put a lens on and a simple circuitry, and they created an imaging software that could be downloaded into their smartphones. And then they were able to run the sample by their smartphones.
To be honest, this hardware team is fire, guys.
Yes.
Fire.
Like, just with that, they were able to quantify the colorimetric signal from the LFA and get the results. So I think if other people would use their approach, it would save so much money.
Yeah, true. Of course, it would save money. And how funny would it be to diagnose yourself at home if you could do that?
Yes, that would be so crazy.
And not just that they were super creative with those tools.
Yes.
They really created a fundament for other teams to build on, I think. Because they also created protocols how to
transform, for example, those plasmid into the bacteria, and then how to isolate the biomarkers that they obtained from that.
Yes, and this is where other teams can build up,
Yes.
And maybe this is the right point to also introduce what they did in their human practice team.
Yeah, because they also were very productive during the project. So they had several projects in the human practice team. For once, they created different educational infographics in different languages even, and also brochures and websites, and they had different target groups, so people of different ages, different backgrounds, and that they did to spread awareness about endometriosis in general and women's health. And then they displayed this info material in several clinic settings. They posted it on different social media platforms, so also on their iGEM account, of course, from Instagram, and also made them available for download at the websites of different endometriosis foundations.
Yeah, and further, they also had a collaboration with the Health Promotion Office at the University of Rochester, right?
Exactly, yeah. And what they did there was that they held a panel that focused on current gender disparities in healthcare.
Which is a huge topic, by the way.
Yes, it is. And they also created a space for students to ask questions about different issues, but it didn't just stop there. They also invited 3 different guest speakers. So one endometriosis specialist from the University of Rochester Medical Center was invited. Also, and that's really interesting as well, a philosophy professor from the university that teaches courses about the philosophy and feminism. So it also gives them another approach to that topic, I think.
It's just a different view on the topic.
Not just the science view, but also the philosophy view. And also they invited a speaker from Columbia University, and the speaker created an app that's called Fendo, and I think she's also affected by endometriosis. And this app allows patients to track and manage the symptoms.
Yeah, so like a menstrual calendar, kind of.
Yeah, exactly, just for endometriosis. So all very important topics.
I think it's always good to have like specialists and experts from different areas of research to come together and talk about things and give just an overall overview and not just one perspective of the problem. You know, that's really cool that they did that.
Yeah, I think so too. Also, they did the same for the menstrual cup design. They didn't just like try it by themselves. They also consulted different menstrual cup companies and stuff. So to really get the best result, and I think they did a great job.
Yeah, and also I think they did a lot in the way of collaborations.
Yes,
that's the next thing. They were like super involved with different teams in that topic.
Yes,
and maybe you can introduce a few.
Yeah, so one thing that really caught my attention was that they had a partnership with the iGEM team in Linköping, so from the same year, of course. And with this team, they were in close contact in regards of wet lab modeling and software. So they had weekly meetings where they discussed areas that were of interest for both teams. And also the team in Linköping utilized their plug-and-play approach as a proof of concept.
Oh, cool.
Yes, they also used it to like obtain their biomarkers, I think.
It's so cool when you can help other teams with your team project.
Yeah.
I think this is what iGEM is about. All like collaboration, like helping out each other.
Yes, they really were into this iGEM spirit. I think they did a great job, especially with Corona. Then of course they participated in several collaboration with other teams. I think there's a page on the wiki because they had many collaborations, and also of course with several foundations that display their material. And also what's another great thing is that they created a biomarker database for other iGEM teams to use. And how this works is that using this database, other teams can search for biomarkers. It's completely free, of course, and if they use one biomarkers, they can just do an entry and they can write about what worked for them, what didn't work, and this makes this whole process of finding biomarkers for projects way easier.
This is really great, and also not just for projects, but maybe also for research in general.
Yes.
Maybe some researcher also goes to that database and tries out.
Yeah, and they don't have to start at zero, so they have a point where they can start. It saves time, I think.
Research is always about sharing information with other scientists. I think this is what makes it easier for all. It's really cool.
Yes, I agree completely. And in addition to that, they also had an accessibility collaboration. And I mean, we looked at in the course of this podcast series, we looked at different wikis, and that's something that I didn't see so often. So they really try to make the science accessible to all people, which is why they started a collaboration with BITS in Goa, IIT Madras. I think they were the one that initiated it, and IISER in Berhampur, and yeah, as I said, of course, the team in Rochester as well. And in this collaboration, they discussed the topic of providing accessibility through language and art. And for that, they also learned sign language phrases. And on the wiki, you can even find a video of someone describing the project in sign language. So it's really well done. And
it's super hard, like to learn it just for the project. I think they put a lot of work in that.
Yes, and then also to show it in video format, that it explains everything for people that might not be so into the topic.
Yeah, and also for people who don't understand the webpage, maybe.
Yes.
Yeah.
So it was really well done. Check it out. I can only recommend it. And of course, they also, because I think it was during the pandemic and stuff, so they to have some fun as well, they also participated in a dance collaboration and video collaborations, and there are even screenshots from that on the wiki as well. So it's really cute. I think you can just see them dancing in the different Zoom video frames.
I think they did it in a very hard time.
Yes.
So for all of our students, it was kind of struggling at one point. We were struggling, and I guess they just coped with that very well.
Yes, they just did the most of the situation.
Yeah.
And they did such a great job.
Yeah.
Yes.
Really. So a very impressive topic here. And I think for everyone listening outside, you should definitely check their page and read a bit into it. And if you're interested in endometriosis, of course, go for it. It's worth. And like always in our podcast, we now want to address maybe related projects to that. And we found out about 2, and we will shortly dive into that. And if you want to find out more, Google the topic.
Yeah. So I think it's really good that endometriosis is that the focus is on endometriosis.
It's very present right now.
Yes.
Yeah.
So it's really important, and so it's no surprise that there are other teams that deal with the topic as well. One team is Paris from 2021. It's called EndoSeq, and it's from the University of Paris-Saclay. And what they did was that they tried to develop a blood test that could be used instead of the laparoscopy. And for that, they tested for the presence and also for the condition of microRNA that circulates in the blood sample.
Yes, because they found out that, or like over the years now, it was clear that microRNA is associated with the regulation of expression of approximately 60% of the mammalian genes, and also could be a biomarker for pathological conditions like in cancer, for example, and especially in prostate cancer and endometriosis conditions, as they are kind of tumor tissue tissue building conditions. So that's what they tried now.
And how did this test work?
I think the test works via signaling cascade. So they use a signaling cascade using 2 Cas proteins, the Cas13A and the Cas13A1, to increase detection sensitivity. Mm-hmm. And as I said, it relies on those 2 Cas proteins, which can cleave different nucleic acids.
That's crazy, because I mean it's a similar topic, but it's a whole another approach.
Yeah, it's just a different view. So they started with using just a different target.
Mm-hmm. I'm really fascinated by the fact that you can use so many ways to do basically the same thing. So I think you should definitely check the wiki out, because...
And also you see here the urge of finding a new diagnostic tool is still very present.
Yes.
And that's why a lot of teams work on that. Yeah, and there is actually also a team this year dealing with it.
So it's this year's teams from Zurich. And since they're still in the process of finishing their project, we can't tell you that much about it right now. But what we know is that they use bacteria as a potential delivery vehicle for the treatment of endometriosis. And here, this treatment would target and influence the unhealthy endometrial cells in the uterus. So it sounds really, really cool. And their goal, of course, is to provide a non-surgical avenue for treating endometriosis. And
so again, it's a diagnostic tool, kind of, and they want to design a diagnostic tool. And yeah, so we have now actually 3 different views presented to you how to use a different way to diagnose endometriosis without using the surgical approach.
I really can't wait for the iGEM Zurich results. So maybe if you're interested in the topic, check the wiki out in a couple of months when this year's competition is finished.
So we are very looking forward to see you at the Jamboree. I would say.
Yes, you will see him there. Right.
So yeah, with that, I think we would finish for today. And as we already said, check out the wikis, get yourself informed, and of course,
follow us on Instagram if you want to.
Yes, and raise awareness for endometriosis.
Yeah, well said.
Yeah, so we are out here. Bye bye. Have
Ciao.
fun.